Good morning, microbiology students. It's Mr. Olson. For those of you that are not in class, I wanted to make a video kind of talking about our macromolecule notes. Let's take a look at those quick. So we have been looking at macromolecules in chapter 2, really what is necessary for life to exist in terms of a chemistry standpoint. And kind of that's where we're going to be at right now. And I wanted to explain these notes for those of you that are not going to be able to be in class. Okay. Again, we're in chapter 2 and we're looking at beyond just the atoms of life, what are the macromolecules needed for life to exist. Okay. So we start with what are living things made of. Okay. And really we can boil it down to pretty much 6 atoms. Okay. There are 6 major atoms that make up the majority of our bodies and cells, and they are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. You'll hear me refer to these as CHOMPs. Okay. 96% of you and every living thing on the planet essentially is made of those 6 elements. Okay. And so those are the really ones that we're going to focus on here. Those 6 elements are going to combine in a bunch of different ways to make these things called macromolecules. Macro just means big, so these are big molecules. Okay. And the 4 macromolecules are carbohydrates, lipids, proteins, and nucleic acids. And as you see these, carbohydrates, lipids is a fancy word for fats, and then proteins, where have you seen carbohydrates, fats, and proteins before? Well, in food, right? We tend to think of those with nutrition, and that's no coincidence, right? If you look at what are the things that you need to put in your body for nutrition, it's probably the stuff that your body's made of, right? That makes sense. So if you ever want to make more cells, make more tissue, whatever, you have to eat the things that your cells are made of, right? That kind of makes sense. And this is kind of what we see here. So these are just some functional groups from organic chemistry, just illustrating that, we're going to see those elements, CHONPS, just in a variety of forms and bonds and arrangements to create really all the molecules for life, essentially. And the rules hold up, right? You look at hydrogen, it's single bonded to carbon, right? Hydrogen wants one more electron, and so it's always going to bond one time. You can see that over and over again. Hydrogen's a great cap molecule. If you have any sort of molecule and it's looking for a bond, hydrogen almost always fills that role. Carbon has 4 valence electrons. It wants 4 more, so it's always going to form 4 more bonds. 1, 2, 3, 4. All right. Oxygen has 6 valence electrons. It wants 8, so it's going to look for 2 more. Right? You see the carbon double bonded to the oxygen there. That's no coincidence. Everybody's trying to get a full outer shell, and we see that. So just as you look at these, don't get too intimidated. It's the same rules that have applied since you took chemistry class. It's based on how many more electrons do you want. That's how many times you're going to bond. The bonds and the arrangements just kind of start to look a little scary, but don't worry about that. Okay. So carbs, lipids, which are fats, proteins, and nucleic acids. Okay. And probably the most important thing here that you'll see over and over is that carbon is the backbone for almost all of these molecules. If you look in the middle, it's almost always anchored with carbon. It's because carbon's unique in that it'll form 4 bonds. That is a really big deal in chemistry. That's essentially, for rules for us right now, that's essentially the max amount of bonds that an element can form is uh, 4. And so that's a big deal. It's like having a Lego that can attach on all 4 sides, right? Think about all the stuff that you could do if you could have a Lego that could attach from any side, right? That you can make all kinds of cool stuff. That's basically how carbon works, all right? So how do you build these big molecules we're talking about, and how do you break them down? We call them polymers. Poly means many, mer means parts. So a polymer is basically something made of many parts or many building blocks, okay? A monomer, right? Monomer is one building block or one part, okay? So a polymer essentially is a chain of building blocks. A monomer is essentially one building block. Okay, how do you build those polymer chains? Essentially, how do you build them? You are looking at, you have a chain here. How do you add new links to your chain? How do you add new monomers to your polymer? It's through something called a dehydration reaction. Well, what do you think of when you think of dehydration? It's a lack of water. And that's what we're seeing here. So I'm going to switch to the pen. It might be easier to see. So you have this polymer here, and we want to add to it. Well, you'll see that it's got a hydrogen on one end that's basically capped this side from bonding, right? Whatever this is, this monomer, it wants to bond with something. Well, hydrogen fills that role most of the time, and so hydrogen ends up bonding to it. Well, once you've done that, you have no way for this thing to ever add new links to your chain, essentially. Well, how do you fix that? Well, you have to get rid of this hydrogen. You'll see on the other side of our chain there's an OH. Okay, that's linking here. It basically is a cap on your molecule. Well, if you want to be able to bond this monomer to this chain, you get rid of the hydrogen cap from this side and you get rid of the OH cap from this side. Well, what do you get when you remove an H and an OH? You get water. So essentially what we say is in a dehydration reaction, you release a water molecule. And you can see that here, okay? What does that do? It basically frees up this part of our monomer to bond, and it frees up this part of our polymer to bond, and you'll see they form a new bond right here. That's how you basically add new links to your chain or new monomers to your polymer to build this bigger, more complex molecule, okay? Let's erase some of this stuff, and let's talk about, now that you know how to build a polymer chain through a dehydration reaction, let's look at how do you go about breaking down a polymer chain, right? How do you break stuff down? Your body's always breaking stuff down, whether it's food or other substances. You guys know that. How do you break stuff down? Well, you add water, okay? Water is that polar molecule. Has an ability to basically weasel its way in between bonds, okay? And sometimes with the help of enzymes, it can do that as well. So if you want to break this chain down, this big long chain, how do you do that? Well, you add some water. Sometimes you have an enzyme involved, but essentially what you do is you break this bond. And when that happens, how do you get it from rebonding? Well, you disassociate your water. The hydrogen goes to this side and caps that in so it doesn't reattach. And then the OH from water goes to the other side and caps that in so it won't reattach. And so water is kind of an amazing molecule. It's super helpful in our bodies. You know, you have to have water. You just don't know what your body does with water. One of the things it does is it uses water to help break stuff down in your body. Okay, it's important. So we call that a hydrolysis reaction. Hydro meaning water, lysis means to break down. And so a hydrolysis reaction is basically using water to break stuff down. Okay. So if we had to kind of summarize that, removing water builds a polymer, adding water breaks it down. Make sure you understand that. All right. Let's look at our first macromolecule. They're called carbohydrates. You've probably heard of carbohydrates before. Really, they're just sugars. Carbohydrate is a fancy word for sugar, and sugars come in different forms, but they're always made of carbon, hydrogen, and oxygen, and typically in this ratio of 1 to 2 to 1. So if you have one carbon, you'll have two hydrogens and then one oxygen. In this case, we have 6 carbons, so you have to double that to get your hydrogens and then single that to get your oxygen. Okay. So you look at carbohydrates, they're building blocks or what we call simple sugars. This is a good example of a simple sugar down here. This is glucose. Okay. Sugars typically come in hexagons or pentagons. We call them sugar rings. Okay. And so we would say this is a sugar ring or a simple sugar. These are the building blocks. You add a bunch of them together, you get a long chain of those sugar rings. That's the polymer form of a carbohydrate. This is the monomer form of a carbohydrate. Okay. What do we use carbohydrates for? They are your main energy source and typically immediate energy or short-term energy, we say. Okay. If you need energy, right away, you got a race in 20 minutes, carbohydrates are a great thing to have. If you need energy in 6 hours, carbohydrates are still a good go-to for that. Okay. They're great for energy, but we also have them. They're a part of cell walls in bacteria. When we start looking at bacteria, they actually are important in bacteria. They're what cell walls are made of, and because of that, we can attack those a little bit differently because we know our cells don't have cell walls. Okay, and plants also use carbohydrates for structural support. You think about celery. Celery has those strings on the back. Those are made of carbohydrates called cellulose, and so those are things that different living things use carbohydrates for. Okay. And we have three types that we look at. We have monosaccharides. Saccharide is just that's a Latin or Greek word for sweet tasting. So we have one sweet tasting, a monosaccharide, made of one simple sugar or one sugar ring. Good examples of that are glucose and fructose, right? Your cells love to use glucose as an energy storage. One of our rules is for your body to be able to use this stuff, it has to be in its simplest form, like a single sugar ring. If it's a complex or long chain, your body will have to break that down ahead of time, okay? Or break that down before it can use it as energy, I should say. We have disaccharides. Di means two, so this is two sugars linked together, two simple sugars. Sucrose, this is the stuff you put on your cereal. It's made of one glucose ring and then one fructose ring. It's the stuff that tastes sweet. Granulated sugar. Milk, that lactose sugar in milk is also a disaccharide, and then maltose is a disaccharide. Okay. We have polysaccharides. Poly means many or many sugar rings together. You have three or more together. That's what we call a polysaccharide. Where do we see these? Starch in plants, cellulose in plants. Starch is what potatoes are made of or pasta is made of, right? Starch. Typically in a lot of plants, they'll store their sugar as this stuff called starch. That way they have it later, maybe over the winter time if you're needing it, or rainy day type energy. Okay, they'll store it as starch for a rainy day when they need it at a different time. And then glycogen is oftentimes how we store polysaccharides in our body. We'll store it as glycogen in like your muscles and things like that. And so when your muscles sometimes need energy, they'll rely on those glycogen stores. Okay. Your body has enzymes to break down polysaccharides into more usable sugars. Okay. I'm going to make a note here quick. I think this is important. So as you look, monosaccharides and disaccharides, those tend to taste sweet. Forgive my mouse drawings or writings here. These tend to taste sweet, right? These are the things you think of when you think of sugar. Polysaccharides tend to taste bland. So pasta, right? Bread, um, potatoes, those kinds of things have polysaccharides in them, and they're bland, right? You don't taste, a pasta noodle and it's sweet. That's because it's in this long chain, okay? And your body doesn't recognize that as something tasting sweet. Things like glucose, definitely sweet. Things like sucrose is a disaccharide, tastes sweet. These sugars are easily used as immediate energy. I think that's also worth knowing. Monos and dis, those are when your body needs energy right away, it's going to look to monosaccharides and disaccharides. Okay, so if you're needing energy 20 minutes before a race, sweet tasting sugars are the way to go. Okay. You'll also hear about people doing what's called carb loading. You've probably heard of carb loading before. That's when you eat a lot of polysaccharides, pastas, grains, breads. What your body does is it builds up a big store of these polysaccharides, these long starch chains. And over time, right, as your body needs them, let's say you're running a marathon and you're out there for several hours, your body will take and break all these bonds between these sugar molecules, and then you can use them as fuel. Okay, it's kind of like trying to start a fire with a tree stump, right? You try and start a fire with a tree stump, it takes a long time, right? It's not easy to start a fire when you've got a big, huge piece of wood. What works better is kindling. You make a little pile of kindling, that stuff will start up right away, and you get your fire, right? Same thing in your body. When it's in this simple form of a monosaccharide, your body can easily break that down into energy and use it. And when it's in a big complex form like starch, it takes your body a long time to break it down. You'll oftentimes see people carb loading days before a race. That way their body has time to break that stuff down into more usable sugars like monosaccharides. And if you look, isn't that what a starch is? It's just a bunch of sugar rings attached together. Well, you break those bonds and you basically end up with a whole pile of usable sugars. Okay. And the last thing I want to mention is any sugar that you don't use as energy gets stored as fat. Okay. So if you eat, some candy bars or things like that, and your body doesn't use those, use the sugar right away as energy, it'll store that as fat for a rainy day. Okay, and it's really easy to convert these over to fat. You look at starches, it takes a while to break these down. And so because of that, it's a little harder to turn this starch into fat because you're slowly breaking these down into useful sugars. When you eat, a candy bar or a bunch of chocolate, you're basically giving yourself a bunch of these monosaccharides. And if you can't use them all right away, your body will then convert those into fat and store those for a rainy day. Okay. There's glucose, and like we said, it's really just carbons, hydrogens, and oxygens, right? All that happens, what's interesting, glucose and sucrose, you'll notice there's an oxygen linking this sugar ring to this sugar ring. If you want to break this down into usable energy, you just snip that one bond. It's not a big deal, okay? We have enzymes to help with that. The one thing they're not showing here is where all the carbons are at. You can see one, this is glucose, C6H12O6. Well, if there's 6 carbons, where's the other 5? Every place where you see a bend or a point, there's a carbon there. So there's a carbon here, 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 and here. And that makes 1, 2, 3, 4, 5, and last one, 6. There's your 6 carbons, okay? Go ahead and erase all the ink on that slide. There we go. All right, let's keep moving. We talked about carbohydrates. I want you to think of those as energy, right? And um, simple sugars like monosaccharides and disaccharides are immediate energy. You look at polysaccharides, those are energy for a little later, okay? Lipids, what are their building blocks? Glycerols and fatty acids, we're going to look at those in just a second. We have different types of lipids that we care about. We have fats, phospholipids, and then steroids, okay? And steroids aren't necessarily what you think of. Your body naturally has steroids in them, like testosterone and estrogen, things like that. And so don't think of, steroid injections. That's not what we're talking about necessarily here, okay? What do we use fats for? Long-term energy storage, right? Those tend to take a long time to break down. So if you're trying to lose weight, you'll notice it probably takes a while. You have to do a lot of exercise to use up that energy, okay? Long-term energy storage, your body will store that for months, years sometimes. And so, think of fats as long-term energy storage, okay? And once you break them down, it releases that energy. Your body uses it to, fuel after exercise or during exercise. And then eventually, you'll see that fat start to get used up, okay? Phospholipids, these are what make up the cell membrane. This is really, really important. We're going to look at those in just a second. And then steroids, things like cholesterol. Steroids typically have this 4-ring structure, 1, 2, 3, 4, but the same elements over and over: oxygen, hydrogen, carbon, right? We said there's only 6 needed for most of life's molecules, and you're seeing that. The steroids have this 4-ring structure. They help regulate hormones. Like cholesterol is one of those. So you need a little bit of cholesterol. It helps with hormone regulation. So don't let people tell you that cholesterol is all bad. You got to have a little bit. Okay. When we look at fats that we eat, there's really 2 types. They're what we call saturated fats. It means that they're fully saturated or full of hydrogen. They tend to be straight molecules, no double bonds, and these are bad fats. So we tend to think of straight and stack and solid at room temp, the S's, okay? Saturated, straight, solid, stack. Bad for you. And then unsaturated fats. These are the ones that tend to be good for you. When you think of fats, these are the ones you want to have in your diet. They're not full of hydrogen, and they're kinked or bent due to some double bonds. Let's look at that. So here's our saturated fat. Here's our unsaturated fat, and you'll notice, if we look at it, you have this carbon backbone. We talked about that. You can see that carbon backbone. And you see everywhere a hydrogen can, it is bonded, right? All along the top and all along the bottom. Hydrogen's everywhere. That creates this sort of linear straight molecule that allows them to stack really well. If you can stack really well, it becomes a problem because they can kind of glue themselves to each other. They're solid at room temp and they want to be solid in your arteries. If they stack, they also want to stack in your arteries. And so these are the ones that aren't good. Animal fats, butter, the inside of an Oreo, unfortunately, right, is a saturated fat. And so these are the ones you kind of want to stay away from. These are the ones that tend to cause problems with your heart and things like that. What you want to have in your diet are what are called unsaturated fats. They're not full of hydrogen. Notice you're missing a hydrogen here, 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 and here. And that's because the carbons have double bonded to each other to get that fourth bond instead of finding a hydrogen. Well, what does that do? It kind of bends the molecule. You see how it kind of goes straight, and it bends up, and it bends down, right? Because of that, they can't really stack, and so they tend to slide past each other. And they stay liquid at room temperature. So that's good, right? Things like olive oil, those are unsaturated fats. Any sort of plant-based fat, those are unsaturated. Those are the ones that tend to be very good for you. They stay liquid at room temp. They stay liquid in your body, and you don't have near the trouble with your arteries and your cardiovascular system with unsaturated fat. These are the ones you want to have in your diet, okay? So remember, saturated bad, unsaturated good. What's the difference? Basically, whether or not you're full of hydrogen or not, and how that changes the shape of your molecule. The other one that is really important, especially for us in microbiology, are phospholipids. These make up the cell membrane. You'll see this lipid bilayer. There's a layer of lipids here, and then a layer of lipids down here. Okay. This is what makes up your cell membrane. You can kind of see they have this head and tail structure. There's the head, and they have two tails. The head likes water. It'll be attracted to water. And the tails are afraid of water. And so what keeps water from dissolving our cells? It really is the tails on the inside, right? They prevent water from just getting in and breaking stuff down and causing all kinds of problems. So we do need that, okay? How do you make a phospholipid? Well, you need phosphate, right? Phospholipid, you need a phosphate group. There it is right there. And you need a saturated fat. There's one there. And an unsaturated fat. Notice how one side is kind of bent, okay? So that's how you make a phospholipid. That's how you make that cell membrane that we know is so important for regulating what goes in and out of the cell. You need a phosphate and a hydrophobic tail, which is made of a saturated fat and an unsaturated fat, okay? So we talked about lipids. That's a fancy word for fats. We talked about carbohydrates. We want to look at proteins now. And proteins, they're a big deal. They essentially do everything. They're made of what we call amino acids. That's their building blocks. And there's 20 amino acids for living things that we care about. I'm not going to make you memorize those 20 amino acids, but I do want you to know there's a variety of them, okay? Amino acids combine to make these things called polypeptides. It's basically a chain, okay? A chain of amino acids. And then once those polypeptide chains start to fold up, that's when you get a protein, okay? Functions, very versatile, really everything, okay? They're important for building muscle and hair. Your hair is made of protein. We have enzymes protein that help speed up reactions. If you didn't have enzymes, you would die. They're actually really, really important. We looked at those in our simulation. You have collagen proteins. They're really important for structural support in your body. Your ligaments and tendons have some collagen. Your bones have some collagen. Your skin has collagen. It gives you the bounce in your skin. You have hemoglobin proteins. They help carry oxygen. That's a big deal, right? You have antibody proteins for your immune system. And so, I mean, it's easier to find things that proteins don't do because they essentially do just about everything, okay? The last part about proteins that I want you to know is this thing called denaturation. To denature means to unfold or unwind, and proteins only work when they're folded up, okay? And so denaturation is a bad thing. How do you denature a protein? Well, heat and chemicals are really the way that we denature a protein. So here's our polypeptide. It's just a chain of amino acids, okay? And eventually, that chain will fold up and make this weird folded up structure. This is a functional, happy, usable protein. This one is not, okay? Now, sometimes if you apply heat or chemicals to these proteins, they will unwind. We said that's called being denatured. And once you go from this happy folded up structure that works to this basically unfolded structure, this protein will no longer work, okay? And so this is kind of why, getting a high fever is a big deal, because if you get too warm, your body's proteins can denature. And if that happens, your body starts to shut down, and it really is a big problem, okay? And so all the proteins in your body, if you get them too hot, it's a bad thing, right? It's going to cause them to denature. And how a protein folds determines its function. Make sure you understand that. Anything else I want to say about that slide? No, as long as you understand denaturation, that really is the biggest thing, okay? The last thing is nucleic acids. They're made of what we call nucleotides. That's their building block. This is a nucleotide right here. They have a sugar ring, a nitrogen base, and a phosphate group. So we're seeing everything but the sulfur, right? We have C, H, N, O, and P. We're seeing everything but the sulfur. That is actually in protein. I'm not going to look at that, okay? So there's three parts: a sugar, a nitrogen base, and a phosphate group. Now, every single nucleotide is exactly the same in their sugar ring and their phosphate group, but they have different nitrogen bases, okay? And so we have like the adenine nitrogen base that makes, when you look at DNA, that's the A, right? We have guanine, thymine, and cytosine. Basically, it's just different nitrogen bases there, okay? Really, the nucleic acids that we care about is deoxyribonucleic acid, also known as DNA, ribonucleic acid, which is RNA, and then this stuff called ATP, adenosine triphosphate. It's got that those phosphate groups on there, the ring, and then the adenine group there. And so it looks a lot like a nucleotide with two extra phosphates, triphosphate, 1, 2, 3. These are really important for energy inside of our cells. When you bust off this third phosphate, this guy right here, it releases a ton of energy in your cells, and that's how you get energy out of food, okay? Our functions for nucleic acids, they're important for storing our genetic material. DNA is important. It's how our cells end up getting energy, and it provides instructions for what the cells are going to do, okay? So nucleic acids are really important. You don't eat nucleic acids. Well, you do, but your body can't take the DNA from a banana and turn it into usable DNA for you. Your nucleic acids come from your parents, typically, right? So your DNA came from your parents. Okay, now the nucleotides you can get from other places and the building blocks to make those, but when you eat banana DNA inside of the banana cells, you don't incorporate that into your DNA, obviously. Okay. The last thing we want to talk about with enzymes is this idea of activation energy. What do enzymes do? Remember, enzymes are proteins, so they can be denatured, right? That's what you looked at in your simulation. And the thing I want to say about enzymes here, I'm going to switch my pen color. It's going to be blue. Is normally anytime you want a reaction to occur in the body, you have to put in some energy. This is symbolizing the energy you put in that's going up the hill, okay? You have to put some energy in to get some energy out of a reaction. What enzymes do is they lower the amount of energy you have to put in. They make things happen faster. I always think of it like pushing a boulder up a hill. That would take a long time to push this boulder up the hill, but if you could do that, you would see a big reaction out of that boulder. It would gain a bunch of speed and cause a bunch of problems when it gets to the bottom. With enzymes, you don't have to push that boulder as far up the hill, and you still get energy out of that boulder, okay? Enzymes lower activation energy, okay? They make things happen faster in your body. Factors that affect enzyme, the pH of wherever the enzyme is at, its environment, the temperature of the environment, and then how much stuff it has to break down and how many enzymes are available. Those things affect your enzyme function. How do they work? You have an enzyme, it has this thing called an active site. It's a special groove or notch in it. And that special groove or notch is what binds to what we call the substrate. It's basically the thing you're trying to break down. So this is the substrate sucrose. It's table sugar. This is the enzyme sucrase, right? What changed? The ACE, right? Usually enzymes end in the letters ASE. And so if you're wondering what the enzyme sucrase works on, It works on sucrose. So use the word to help you figure out what it works on. So you have the active site, the special binding site for the enzyme. It then locks in with your substrate. You can see how it's kind of locked in, it fits perfectly together. The enzyme then breaks the bond between those two sugar rings. Let's erase this. The enzyme then breaks the bond right here, this oxygen. It breaks this bond right here with the help of water, right? This is called a hydrolysis reaction. We're using hydro to break stuff down, lysis, and you get two new things. You get this glucose and this fructose. And so the enzyme sucrase breaks down a sucrose molecule into fructose and glucose, and these are called your products. So you have your active side of your enzyme, you have your substrate, which is the thing getting broken down. It locks in to your enzyme at the binding site or the active site, and then the products get released, and then you get these new things. And enzymes can do that over and over and over again, right? You don't ever use up the enzyme. The enzyme, notice, goes back, and it's ready to go break down another sucrase again, okay? So make sure that you look at this graph, okay? Looking at enzyme activity. Remember, this is in Celsius. So if you look at which enzyme works best at lower temperature, this is the optimum working temperature right in here for enzyme A, and this is the optimum for enzyme B. So obviously enzyme A works better at lower temp. At what temperature do both enzymes have the same activity? It's where they intersect, right here, maybe 48 degrees Celsius. 48 degrees Celsius. And which enzyme would be most likely found in the body? This one's tricky. Kids want to look here, but this is in Celsius. This is about 37C right here, and that's actually human body temp. So enzyme A actually would work better in the body, okay? So hopefully that helped kind of square away enzymes for you, or not just enzymes, but macromolecules in general. I know the video is long, but if you have questions, let's make sure we talk about it. Otherwise, we'll see you guys on Wednesday and Thursday.